Okay, so Assalamualaikum warahmatullahi wabarakatuh and hi. So today, before we learn about the new topic, let us have a thinking corner. First, please look at your name. When is the last time you cut it off? How fast is it growing? Have you ever noticed? Can you see the nails growing? So, can you tell your own imagination on how the nails are growing? All this while, how you think your nails are growing? And this is the hair growing. Is it the same with the process of the nails growing? So, today we will learn about the cell division. So first, cell division is divided into two, which is the karyokinesis and also cytokinesis. So, karyokinesis is the division of the nucleus. Okay, as you can see here, while cytokinesis is the division of the cytoplasm, which is the split of the cytoplasm into two daughter cells. So first, in organism cell, we have somatic cell and also reproductive cells. So what is somatic cell? Somatic cell is the body cells other than the gametes. Okay. So the somatic cells produce through mitosis process, and it contains diploid number of chromosomes, which each cell contains two sets of chromosomes or 2n. In human somatic cells, 2n is equal to 46. So if you can see the diagram here, okay, the diploid we have 22 here plus 23 pair. Okay, 23 pair meaning that we have 46 chromosome, because the chromosomes, the chromatic here, it will undergo replication to form a complete chromosome. Okay, each pair have two complete chromosomes. So for the reproductive cells, the gametes are reproductive cells. So gametes are produced through meiosis process, and it contains haploid number of chromosomes, which each cell only have one set of chromosomes, and in human gametes, n is equal to 23. If in the somatic cells you have a complete 46, okay, 23 pair, but in human gamete for the reproductive cells, you only have 23 chromosomes, not 23 pairs. So first, in diploid cell, one set of chromosomes originate from the male or the parent or paternal, and another set is from the female or the mother or maternal chromosome. So if you can see here, one is from the paternal or papa, we call it here. So we know it's from the father. So maternal M it stands for mama, so you can know it from the mother. So the paternal will undergo replication, as I had explained before, and it will become a complete chromosome. Okay, so the both maternal and paternal have same structural characteristic and are called homologous chromosomes. So here, these paternal and maternal, okay, when it undergo replication, these two chromosomes we call as homologous chromosomes. And if you can see the diagram here, okay, for example, the blue one is from the father or the paternal, the red one is from the mother, meaning that in complete human cell, you will have one from the father and also one from the mother. And if you can see how it said that the maternal and paternal have same structural characteristics because each of the line indicates the same gene. If you look at here, okay, you can see that the line is we call band actually. This is the first band, the second band, the third band. So if you can see the width also is the same for both the maternal and also paternal. The blue one is the paternal, the purple one is the maternal. So as you can see, all the sequence of the bands or the lines, the width is all the same. So the first line for both maternal and paternal, for example, is the color of eye. That's why the width is the same. If it is not the same, the first of the paternal, okay, will have different with the maternal. For example, the first for the paternal is the color of eye, but for the mother is the hair type. So the width of the line or the band will be different. Next, chromatin is a chromosome that looks like long thread. So before we form chromosome, uh, if you can remember in the subtopic nucleic acid, the chromatin is the one who made up the chromosome. Okay, it will form the chromatin structure between the folded of the histone with the DNA. Okay, this is the structure of the chromatin. Long thread, which when it condenses, it will form a chromosome that we can easily see. So the sequence of events that involve DNA multiplication and cell division to produce two daughter cells. So that is the aim of the cell cycle. It will produce two daughter cells under a series of DNA multiplication and also cell division. So cell cycles is divided into two. First is interphase and also the second is the M phase. For interphase, it is made up of the G1, S and also G2. So here is the G1 phase, okay, it will undergo the G1 phase, the S phase and the G2 phase, and it is the longest phase in cell cycle. If you can see the gap here, you can see this is a long gap and this one is the shorter gap. Okay, so we will go first with the interphase. For the G1 phase, okay, at the G1 phase, the cell will grow, meaning that the cell will produce the organelles such as mitochondrion and also endoplasmic reticulum and the others, okay. And protein that should be used by the cell will be produced or they will be synthesized at this phase. And the nucleus will look big and chromosome is still in the form of chromatin, okay. And next it will go to the S phase. So S phase is the phase where the DNA synthesis occurs. Okay, the DNA in nucleus is replicated. If you can see before this, the diagram that I show you, right here. So here you have two, right? This is incomplete chromosome or incomplete sister chromatid, but here you have this one is the complete. Okay, this one you will undergo replication to become this. So that's how it explains each chromosome will multiply into two identical chromosomes called sister chromatid. Okay, so this is the sister chromatid. This one, the two is the same. Okay, and both chromatid have the same copy of DNA and join at the centromere. Centromere is at the center. So the cells continue to grow and remain active metabolically in G2 phase, and the cells will gather energy and make final arrangement before it enter the cell division. So after this stage, we will enter the M phase. But remember that the interphase is the longest phase. Okay. So this is how briefly the interphase. Next, we will enter the M phase. For M phase, we have the mitosis and also the cytokinesis. So this is the mitosis. The first stage is prophase, or we can remember it as P-mat. P for prophase, M for metaphase, A for anaphase and T for telophase. So for prophase, in nucleus, chromosome starts to shorten and thicken to make it visible under light microscope. Okay, and then the chromosome is made up of two identical traits called sister chromatids. So this one is chromosome. Okay, it will undergo replication, become sister chromatid. This one is one sister chromatid. This one is the second sister chromatid because it is the same. Two sister chromatids will make up a chromosome, a complete chromosome. So both sister chromatids join at centromere. So here is the centromere, and nucleus membrane will disintegrate and nucleolus will disappear. Okay, because it wants to be visible under light microscope. So the centrioles move to opposite poles and spindle fiber starts to form. So this is the centriole. Okay, centriole will start to form the spindle fiber and it will go to the opposite form. So this is how the briefly the prophase occur. Okay, the spindle fiber move to the opposite pole. The centriole move to the opposite pole. The spindle fiber starts to form. Okay. So next is the metaphase. The centrioles are at opposite poles of the cell. Just now when it moved to opposite pole, now it have arrived. Okay. When it arrived, the spindle fiber maintain chromosome at equatorial plane. How? The spindle fiber attached to the centromere of the chromosome, and it stay at the center of the cell. The chromosome will align in single row on equatorial plane. Equatorial plane is the center of the between the two poles, and the metaphase end when centromere begin to divide. If you can see here, it make a single line. Okay, a single line at the metaphase. Metaphase plate is also at the equatorial plane. So for the anaphase, the centromere divide into two and sister chromatid starts to separate. Okay, so in anaphase, just now when you make a straight line, okay, at the center, at the equatorial here, when you enter into anaphase, the centromere will be divided. Okay, it will split into two, follow the sister chromatid. Okay, separate, and then the spindle fiber will shorten and contract. So spindle fiber will shorten, thus it will pull the sister chromatid towards the opposite pole or towards the centriole. So anaphase end when chromatid arrive at the pole. Okay, in anaphase it have not arrived; it's just been pulled towards the opposite pole. So here is how it happens. So the last TMAT. So T is for telophase. When chromatids arrive at opposite poles, they are now called daughter chromosome because they already have complete site at each pole, and they will become two daughter cells. Okay, so each pole contains one set of complete and identical chromosomes. The chromosome shape again as chromatin. Okay, chromosome will form chromatin, and nucleoli and nuclear membrane will be formed back, and telophase stage will be followed by cytokinesis. So this is how the process happens. Okay, so can you think where did the mitosis occur in plant? Okay, where did it occur in plant? You can write your answer in the comment. So next we will look at the differences between the mitosis and the cytokinesis in animal cells and also plant cells. So first, plant cells do not have centrioles, but it can form spindle fiber during mitosis. And cytokinesis is the division of the cytoplasm that happens immediately after the nucleus is formed at the end of telophase. So after telophase, cytokinesis will happen to divide the cytoplasm, making two daughter cells. So we have the comparison between the animal cells and also the plant cells. First, let's look at the animal cells. The plasma membrane constricts in the middle of the cell between the two nuclei. You can see that the constriction of the cytoplasm in the middle of the cell or between these two nuclei. So the microfilament at point of constriction will contract, causing the cell to constrict until it splits into two daughter cells. So here actually in the middle of between the two nuclei we have microfilament. So microfilament will start to contract, okay, making the cytoplasm to constrict automatically, and thus it will split into daughter cell. Okay, as you can see the cytoplasm is totally been split. And for the plant cells, the cytokinesis begins when the formed vesicle combine to form cell plates at the center of the cell. If you can see the membrane-bound vesicle, okay, it will combine at the middle of the between the two nuclei. So the cell plate will expand outwards until they combine with plasma membrane. First, the membrane-bound vesicle will become cell plate. Okay, it will grow until it touch the plasma membrane. Okay, it touch at both sides and then it will completely separate the two daughter cells. So at the end of cytokinesis, the cellulose fiber are produced by cells to strengthen the new cell wall. After the cell plate have completely touched the two plasma membrane, forming new cell wall, the cell wall will be covered with the cellulose fiber, okay, to strengthen it. Then only the two daughter cells are formed and each has diploid condition. So this is how the cytokinesis happen in animal cells, while this is how the cytokinesis happen in plant cells. In animal cells, it will constrict until it will split the two daughter cells, okay? While for the plant cell, it will grow the cell plate until it divides the two daughter cells. Okay, this is also to show the differences between the cytokinesis of the animal cell and the plant cell. Okay, it will microfilament will constrict and form cleavage furrow is actually the constriction point. Okay, and it will split. Also, the plant cell will form the cell plate from the vesicle, and it will form new cell wall. So, your task is to find and list out the necessity or the importance of mitosis. Also, gather the information on the application of mitosis in the stem cell therapy. Meaning that you need to gather information of the stem cell therapy, how did it related to mitosis, and we will discuss it in group, which I will select few students to explain and present it. So that's all. Thank you.